here's where we left off yesterday. So if we were to number these steps, this step, glycolysis, would be like step 1. Okay. Step 2, this citric acid cycle, would be this one right here. All right. Now we've got to go to step 3. So step 3, I'm going to zoom in just real big. So if you were to draw it on a piece of paper, ooh. I feel like there's a strong possibility you'll have a quiz on this tomorrow. Okay. So I'm still looking at, so the name of this step, all right, is called oxidative phosphorylation. Okay. Location is going to be the inner membrane. So the inner membrane of the mitochondria. And so what I've got here is I've got a couple molecules. I've got these proteins right here. I've got 3 of them. Okay? These proteins span across the entire length of the cell membrane. So these proteins, they're called transmembrane proteins or integral proteins. They're the ones that span across both sides. So it can go from the intermembrane space right here to the matrix of the mitochondria right here. So it can move stuff between the two. So I've got those, and then I've also got some other membranes that are only, or proteins that are only on the side right here. Okay? We had a name for those proteins that are on the side. Peripheral proteins. All right. So I've got these things right here, and then I've got one last molecule that goes right here. Okay. This is a big molecule right here. It's an enzyme, and that enzyme has a name. It is called ATP synthase. Okay, ATP synthase. So now what happens in this whole thing? So I've got these little transmembrane proteins. Something's going to come in here. All right. So something's going to come in and something's going to go off. What comes in right here is NADH. Notice anything different? NADH enters right here. What do you think comes off? NAD+. It's the opposite of everything we've seen so far. All right? And then on this one, all right, I've got something that comes in and something that comes off. For this one, FADH2 comes in, and what comes off is FAD comes off. Okay? On this one, yes, and there's a reason why, and I'll get to that in a second. So when it gets here, it does something, all right? You remember what we said the whole job of NADH and FADH2 was? They're electron carriers. So their whole job was to carry electrons to the electron transport chain, which has another name called oxidative phosphorylation. So now that this is going to get here, it's going to kick something off. It's going to kick off a hydrogen. It's going to kick off a hydrogen. Okay? So I've got H+, 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 H+. So I've got all these H pluses out here now. So we have a very high concentration of them. Do we like it when we have a very large amount of something that's in one area, or do we like it to be spread out? We like it when it's spread out. So now that I've built this real high concentration of hydrogen ions on the outside, it doesn't like that. And so we've got this molecule called ATP synthase. It's kind of going to be like a turbine in a dam. Have you ever seen what the inside of like the Hoover Dam looks like? So you've got these turbines in there, and so you've got all this water that's a high concentration on one side, low concentration on the other. As it moves through that, it turns these turbines and it generates electricity. As these hydrogens, it doesn't like having a high concentration of hydrogens on one side, so these hydrogens are going to flow down that concentration gradient. As it does that, it's going to turn this kind of like a water wheel. And what that's going to do, it's going to provide enough energy to take ADP plus inorganic phosphate. And what do you think it turns it into? To ATP. So it generates ATP, and it leaves me with these hydrogens down here. Okay, so It brings some hydrogens down here. But now I've got to do something with those hydrogens. All right? So you remember in cellular respiration, we said we start with glucose and oxygen, and we end up with carbon dioxide and water. Anywhere in this whole process that we've looked at, have we seen oxygen come in by itself yet? We haven't. Oxygen doesn't come in until at the very, 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 very end. And nowhere in this have we seen water get produced yet. So now what's going to happen, I'm going to eventually collect 2 of these hydrogens, so 2 of these H pluses, and combine them with 1/2 of an O2 molecule. And what do you think I make? I make water. Okay? Think back to chemistry, electronegativity, and periodic table trends. Which part of the periodic table has the most electronegative atoms? The top right. So that top right corner is the most electronegative atom. So if I look at a periodic table, is there any element up there that we've been dealing with on this? Oxygen, as Seabass says. Oxygen. Meaning oxygen is very electronegative, meaning what does it really, really want? Oxygen really wants electrons, all right? So this whole process happens because we remember we started with a molecule that had a very high amount of electrons. Do you remember what that molecule was? We started with glucose that has a lot of them. This whole process is about taking those electrons from glucose, shuffling them around, and eventually the final electron acceptor is going to be oxygen. And in doing so, it's going to generate all these ATP. Okay? So we've got to look at our net products now. So net products. No. Okay? Eventually what would happen, if you looked on a molecular level, okay, you've got these 3 proteins. What would actually happen on that is you would have, when it kicks off this hydrogen, it also kicks off an electron. So this little electron would come off, and I'd write it like E minus. It would come off, and it would go here, and it would go here, and it would go here. And then eventually that electron would kick off here, and it would combine with water. So this electron, see these little peripheral proteins right here? What they actually do is they sit there and bounce back and forth. So it would sit here and just go back and forth, back and forth, back and forth between these 2 things. If you ever consume cyanide, cyanide stops that peripheral protein from moving. Peripheral protein can't move electrons back and forth. Can't move electrons back and forth. I can't make ATP. You will eventually die because you can no longer produce ATP in your body. That's what cyanide does. Cyanide inhibits this molecule right here. Okay, that one that we just circled in yellow. All right, and so now we've got to look at our net production of all these things in all of our steps. So we're going to make this big table that we have. So I'm going to label them step 1, step 2, and step 3, and I'm going to say ATP. I'm going to say not that one. I'm going to say FAADH2, and I'm going to say NADH. Okay. So let's make a little table now for this. This is what was on the board when you walked in and you guys started freaking out. Like, what the heck, man? Okay. So if I look at these, in glycolysis, how many ATP did I make? 2. In glycolysis, how many FADH2s did I make? None. And how many NADHs? 2. In the citric acid cycle, how many ATP did I make? 2. How many FADH2s did I make? 2. And NADHs I made 8. Okay? Looking at this step right here, how many NADHs did I produce? How many NADHs did I make? In step 3, so step 3 that you see right here on the board, how many NADHs did I make? None. So right here, it would be none, because I didn't make any NADHs, I actually broke them down. How many FADH2s did I make? None. I actually broke them down. All right, so now what we've got to look at, the ATP. This is where it gets a little tricky. This one you cannot just look at it and count, okay? So remember right here, NADH comes in, and it drops off that hydrogen, drops off those electrons. Every NADH, so, I'll do it right here. Every NADH that I made earlier equals 3 ATP. Every FADH2 equals 2 ATP. Okay? How I remember it, if you look right here, NADH comes in at this first protein on the left. So right here, right here, right here, there's 3. NADH can produce 3 ATP. If you look at FADH2, it comes in at the second protein, so you've got here and here. So it can produce 2 ATP. So I made total amount of ATP or NADH I made. Sorry, NADPH is in photosynthesis. Total amount of NADHs I made in all of this right now is how many? Total that I've made so far?
8, 10.
10. If every NADH can give me 3 ATP, how many total ATP can I make?
30.
So I'm going to put NADH equals 3 ATP times 10 equals 30 ATP. And how many FADHs FADH2s did I make? I made 2 total, so 2 ATP times 2 equals 4 ATP. Cellular or electron transport chain can make 34 ATP. You made 2 of them during the citric acid cycle, 1 of them per pyruvate. So if I look right here, it would be right here. I would have FADH2, but remember there's a second pyruvate. Okay? All right. Does this make sense?
Yeah.
Okay? So the third step, it's called oxidative phosphorylation. You're probably wondering why on earth did they give it this big, complicated, stupid name? If you look at it, and you remember yesterday when we talked about oil rig, remember we said RIG, oxidation is losing, reduction is gaining. Okay? If I look right here, the term oxidation means to lose electrons. NADH, does it lose one or does it gain it? NADH loses it. So NADH gets oxidized, and if something gets phosphorylated, do you remember what it means? I add a phosphate to it. So NADH gets oxidized, it generates this concentration gradient of hydrogen ions, which are going to flow through ATP synthase, which provides the energy needed to take ADP and phosphorylate it into ATP. It's called oxidative phosphorylation because I oxidize NADH in order to phosphorylate ADP to make ATP. That's where that name comes from. Okay? So, yes, ma'am.
You know, you draw like those little H's?
The H's?
Yes.
Yep.
Do we need to have that exact amount?
You don't have to have that exact amount as long as I can see you have a few. I think on tomorrow's, I think I put 5 lines. Because I'll put a line everywhere that I want something. I put 5 lines there. Okay? Any other questions on this before I stop this?
We're getting everything in black, right?
You are getting, tomorrow you are getting everything in black.
Including ATP synthase?
Including, oh yeah, ATP synthase will be there also. I'm going to post after this, when I post this online, I'm going to post a little document that's going to have this whole thing, but everywhere I want an answer, I have a blue mark. That way you guys can practice on your own. Okay?
You're not going to have the net products, right?
I won't have net products, I'll have little blue lines in each of those. Okay? Oxygen exists in a diatomic molecule. Oxygen never exists by itself. It always exists in a diatomic molecule. So I'm not going to add 2 hydrogens in O2. I'm just going to add 2 hydrogens in half of an O2 molecule. Because oxygen's never by itself. Okay. Ready?